Module 5, Objective 8, be able to describe the neurotransmitters and neuromodulators discussed on the following slides, offering examples of each. We have two classes of neurotransmitters, excitatory neurotransmitters and inhibitory neurotransmitters. Excitatory cause a depolarization of the postsynaptic membranes, and that's going to promote action potentials. Other neurotransmitter, the other class, is inhibitory, and they cause a hyperpolarization, which suppresses the action potential. If we go back to that same sort of graph of transmembrane potential that we've seen before, so if we're sitting at rest, an excitatory is going to cause a depolarization. And of course, we know that that depolarization is strong enough to reach threshold, then you get the formation of an action potential. So that promotes action potentials. Inhibitory neurotransmitters produce a hyperpolarization, and they're going to actually take the transmembrane potential further away from that threshold, and that's going to help suppress action potentials. The effect of the neurotransmitter on the postsynaptic membrane depends on the properties of the receptors, not the nature of the neurotransmitter. So it's the properties of the receptors that determine whether it's excitatory or inhibitory. And if you look at the major classes of neurotransmitters, some of them are amino acids, some are neuropeptides, even dissolved gases such as nitric oxide or carbon monoxide can serve as neurotransmitters. So chemically, there's a fairly good range of classes of neurotransmitters. In terms of the biogenic amines, there are two that we'd ask you to know. The first one is norepinephrine. These are released by the adrenergic synapses. These are synapses that release norepinephrine or even epinephrine. They tend to be excitatory, so they have a depolarizing effect on the transmembrane potential, and they're widely distributed within the brain and portions of the autonomic nervous system. Dopamine is another central nervous system neurotransmitter. It can be excitatory or inhibitory, just depending on the receptor. And dopamine is pathologically associated with Parkinson's disease or with the euphoric feelings that one gets through the use of cocaine. So here are just two examples of amines, of biogenic amines, which are two neurotransmitter, which happens to be biogenic amines. The other two biogenic amines worth noting are serotonin. This is a neurotransmitter of the central nervous system. It's going to affect also attention and one's emotional states. There are lots of depression-type medications that target serotonin. And those are going to affect one's emotional states as well. The other one is gamma-aminobutyric acid, or GABA. GABA has an inhibitory effect, so it's going to cause a hyperpolarization at the postsynaptic cell. And we still don't know a lot about it. So the functions of GABA within the CNS are still not well understood. While there are upwards of 50 or so identified neurotransmitters, I'm only asking you to know these four in addition to acetylcholine. Neuromodulators are chemicals that are released by the axon terminals that alter, and they're going to alter either the rate of neurotransmitter release or the response by the postsynaptic cell. The effects of neuromodulators are long-term and they're slower to appear. They involve multiple steps or intermediary compounds, which we'll see an example of that shortly. And they're going to ultimately affect the presynaptic membrane, postsynaptic membrane, or both, just depending on the neuromodulator. They tend to be released alone or with a neurotransmitter. Examples of neuromodulators, the neuropeptides. These are just small peptide chains that are created or synthesized and released at the axon terminal. Opioids, they are also neuromodulators. They bind the same receptors as opium and morphine, so they have strong pain relieving types of properties. Classes of opioids include the endorphins, well, the endorphins are up there, the dynorphins, as well as the enkephalins. Neurotransmitters and neuromodulators may have a direct effect on the membrane potential, either by opening or closing chemically gated ion channels. So examples of these would be acetylcholine, glutamate, aspartate. They may also have an indirect effect through something that we call G proteins. And we're going to look at G proteins shortly. And they come up later in AMP2, so learn them now. Examples of indirect effects include epinephrine, norepinephrine, dopamine, serotonin, histamine, as well as gamma-aminobutyric acid. The third relationship is these neurotransmitters and neuromodulators may have an indirect effect via intracellular enzymes. These are going to be lipid-soluble molecules, such as nitric oxide or carbon monoxide. These can go directly through the cell, since they are, that's a really poorly shaped nucleus, but these can go directly through the cell membrane, since they are lipid-soluble, and they're going to affect enzymes intracellularly. Usually they turn them on. So in terms of direct effect, this is what we know best. We know acetylcholine binds its receptor, which is a chemically gated sodium channel here. It causes that receptor to open, and upon opening, sodium then can move in and depolarize the postsynaptic cell. That is the direct effect, and we know this one well from modules 4 and earlier, even in this module. Indirect effects via G proteins. Again, we're going to really just briefly introduce G proteins. They're going to come up again later on. So for now, for a neurotransmitter to bind a G protein receptor, this is an inactive G protein receptor. So here's our G protein, it's inactive, and the binding of the neurotransmitter to the receptor causes the activation of what we know as G protein. And that's going to start a cascade effect. It's going to start a domino effect. And we see that activated G protein is usually going to create a second messenger. So the neurotransmitter was the first messenger. We've now, through the activation of a G protein, we've created a second messenger. And that second messenger is going to play the role of opening the ion channel to allow the depolarization or hyperpolarization event, what have you. So that is the G protein. Epinephrine, norepinephrine, dopamine, histamine, Gaba, these are all examples of neurotransmitters that have an indirect effect via G protein. So again, more to come on the G protein saga, but for now, if you have this basic understanding, I think we're okay. Lastly, indirect effects by intracellular enzymes. And again, for these, you're going to need lipid-soluble gases, usually, such as nitric oxide, carbon monoxide. They simply diffuse through the plasma membrane. They activate an enzyme, which produces, again, a second messenger. And that second messenger is going to do things like open ion channels, or it's going to change the metabolic activity within the cell. So examples of all three types.